Hello, in this video I'm going to talk about meiosis. So meiosis is like mitosis, but mitosis you can say occurs in your toes. It's where all your body cells just divide and produce identical body cells. Meiosis occurs in your O's, your ovaries, or your O's, your huevos, your testicles, or to produce sperm and egg. So remember, your chromosomes in every one of your cells exist. There's 46 of them, but they exist as 23 pairs, 23 homologous pairs. Homologous means the same. They're the same size, they have the same genes on them, but they might have different versions or alleles of those genes, right? So meiosis is the process of taking those 23 pairs of chromosomes and separating them into a set of 23 and a set of 23, which will then make the sex cells, which through fertilization can form 46 again. So let's look at this diagram here. It shows what's happening. So you're conceived, you're a fertilized egg, right? You spend all of your life growing and developing through mitosis. Cells divide and getting bigger, right? Then couple cells in the sex cells, right, in the reproductive organs undergo meiosis and they produce sex cells. So they divide and reduce the number of chromosomes from 46 to 23, producing what we call haploid cells. Haploid cells have half the chromosome number as diploid cells. Diploid is where all our chromosomes are found in those homologous pairs. Okay, those sex cells then unite and produce a zygote, and that cycle goes on and on. So some terms to just remember. We talk about an N number. An N number is the number of different chromosomes in your body, of which we have 23. 2N is those 23 pairs. So the 2N, we talk about the total number of chromosomes, 46. N is the haploid number, one of each pair, which we're producing when we make sex cells. Meiosis is the process of taking body cells with 46 chromosomes and producing 23. Fertilization is the process of taking those 23 and 23 in the sex cells and making 46 chromosomes again. Okay. And then meiosis and differentiation is the process of growth and differentiating development as you grow through your life. All right, so most animals spend most of their life in this diploid stage, and that's really good because we have about 10 or 12 lethal alleles, and if we only had one of them, we'd be dead. But because we have all of our genes in pairs, we usually have a normal dominant allele that prevents us from dying from those 10 or 12 lethal alleles. It's a little different in fungus, and some algae where they spend most of their lives in the haploid state. Okay, so they're going to be haploid most of their lives. They're going to undergo fertilization and then quickly undergo meiosis to produce more haploid organisms. So fungus, mushrooms, and so forth are predominantly their life cycle they spend in that haploid state. And then some plants like moss and very simple plants and other algae spend equal or comparable amounts of times in both the haploid and diploid stage. So not all organisms undergo what we go through, a very brief time as sex cells and a longer period of time as diploid cells, just a sort of comparison of different organisms. All right, so overview of meiosis, right? We start off with a homologous pair of chromosomes. There they are. One you got from your mom, the red one maybe, and the blue one you got from your dad. Before any cells can divide, we have to replicate the chromosomes. So there we have sister chromatids on each. And since we have two sister chromatids and two sister chromatids, sometimes we call this set of homologous replicated chromosomes tetrads, tetra meaning 4 sister chromatids. So the first step in meiosis is we separate the homologous pair. Okay, this is different from mitosis, where we just separated these sister chromatids on all 46 chromosomes. Just the homologous pair gets separated there. Okay, and then we're going to separate the sister chromatids in meiosis 2 to produce 4 haploid cells. Okay, so that's basically what happens. This is kind of a more detailed view of that. One important thing that happens when we go from interphase to prophase 1 of meiosis is, notice we form these homologous pairs and the chromosomes are kind of sticky. They stick together. And then when they line up at metaphase of meiosis 1, and then we pull them away, notice we've seen some information switch. What's called crossing over has occurred. And that's really important because that increases the genetic variation within our sex cells and makes us even more unique from our siblings or from different offspring produced from the same parents. Meiosis 2 basically is similar to mitosis in that we have two haploid cells from meiosis 1 because they only have 2 chromosomes. We have to separate the sister chromatids. So we're going to go through basically the same process of lining them up at the equator and metaphase, anaphase, pulling them apart, and then dividing those 2 cells into 2 more cells each for a total of 4 cells. So there we go, there is meiosis. Now how does this compare to mitosis? Remember mitosis, if we had those 4 cells, we'd line them up at the equator and separate them into 2 diploid cells, each with the same number as the original cell before the chromosomes replicated. In terms of meiosis, we're going to line them up by pairs, and you can see there's a little crossing over occurring in prophase 1 of meiosis there. When we separate the pairs, we have now 2 chromosomes each, so we have the haploid number, but then we have to separate the sister chromatids in meiosis 2 to get a total of 2 chromosomes in each, as compared to the original 4 chromosomes in this sample cell. Okay. One thing between males and females, males tend to go for quantity in terms of sex cells they produce, females go for quality. So in terms of males, we have the cell division followed by cytokinesis, dividing up the cytoplasm evenly and producing 4 smaller but evenly sized sperm cells. When the egg divides, we see that the chromosomes divide in the exact same way as the sperm cells, but cytokinesis does not occur. Splitting of the cytoplasm remains around one set of cells, right? And what that means is that we have one larger cell. So this is because the egg's got to travel that 10-day journey from the ovaries down the fallopian tube. If it gets fertilized, it's got to kind of take care of itself and be able to kind of sustain itself until it gets embedded in the uterine wall and can get those capillaries growing in towards it so that it can kind of be fed by the uterus of the mother, right? So it's kind of built for a longer journey. Sperm, they get ejaculated, they swim around for a day or two, they run out of energy and they die. We produce lots of them. They're like little foot soldiers. They're highly disposable. Eggs, females only produce several hundred of them through their life, releasing once a month. So they're, again, a little bit more sort of valuable. They're larger and we produce fewer of them, and by mid to late 40s, women have released all their eggs and they're done reproductively. There you go. So again, just to look at that crossing over step in a little bit more detail. So in prophase 1, right, as those homologous chromosomes pair up, they stick together. They're like 2 kind of sticky, I don't know, pieces of bread dough. And then when you pull them apart abruptly, you can see that you get some of these segments crossing over, and that increases genetic variation. That's good, but it's also important for linkage. When we talk about traits that are linked, meaning they're found on the same chromosome, oftentimes we'll see linked traits get unlinked. How do they get unlinked? Well, a crossing over can occur so that we can see that maybe 2 traits, let's say red hair and freckles, which are usually found together, get unlinked, and now we see someone with, you know, dark hair and freckles, or we see someone with blue eyes and dark skin, because typically linked genes that we see together get unlinked through this process of crossing over. We can actually use this when we get into genetics to study linkage maps and figure out how close different genes are by how frequently they cross over. And we'll get into that later. All right, but there's a good example of another crossover occurring, and we can see how likely they are based on how close those 2 genes are. And again, the last thing about meiosis that makes it really important is that it really adds a lot of genetic variation. Each of those sex cells is different, right? We have 23 pairs of chromosomes. 23 you got from your dad, 23 you got from your mom. So the different combinations of those 23 is actually 2 to the 23rd. That's about 8.3 million. That's a lot of different sex cells each person produces. And that makes it very rare that you and a sibling will be genetically identical unless you're identical twins. And that's really good, again, for variation and producing diversity within populations, and making sure that no matter what environmental change happens, some in the population will have the right mix of genes to be able to survive and endure. Okay? So that is it for meiosis, and I'll see you next time. Bye.